Thank you for coming. It's a pleasure to see you here. And we are going to talk about immune receptors. And probably I don't need to give introduction to this audience. You know better than me 10 times what does mean immune receptors and how they organized. What I would like to say, it's an extremely interesting biological system, immune receptors, because just look from genetic point of view, you have complexity of all, at least several hundred million of different T cell or B cell receptors constantly present in circulation in our body. And if you think about this number, it's really enormous. Compare with the number of the human genes, 20,000. We are talking about here about several hundred million. So at least 10,000-fold more number of genes for T cell receptor and B cell receptor than number of the human genes. Because I'm a technology guy and I try to develop different technologies all my life, just think of how to do quantitative profiling for the complexity of the genes, 10,000 more than number of the human genes. It's number one technical question which we will try answer today. Second problem, if you think about the system, it's a universal diagnostic system created by nature, and nothing in the scale you can compare with any other diagnostic assay available right now. It detects any practically pathological molecules or foreign antigens which present in our body. Why we don't use it as a universal diagnostic system? Simple answer is very simple. We don't know actually well if we can sequence T cell receptor or B cell receptor, but we don't know what they recognize and how they recognize target antigen. If we solve this problem, in this case it will be the next generation of universal diagnostic tool. And let's hope young people who are present in this room will be already at the beginning of this revolution. They will do it. Okay, let's go to technology. In order to facilitate these studies, we developed three technologies shown here on this slide. And first of them, it's adaptive immune receptor profiling assay, which is bulk profiling in cells or in RNA or DNA purified from cell population, for example, PBMC or tumor sample or any pathological organ. And our goal is to screen of activated immune receptors. It can be used for, and we're going to discuss how these tools can be used for screening with peptides or vaccines. Second assay, it's very similar to first one, but it's designed for single cell analysis. In this case, you sort cells in 96-well plate, and you can get full-length chain pairing information for each receptor and expression of 30 marker genes. And last one is completely clear artificial cell system, JORCAT and FAT reporter cell line, and with K562 HLA as antigen-presenting cells, which allow to validate immune receptor, T cell receptor, and peptide interactions. So, in order just to give you an idea how it's working, it's probably one of the most comprehensive assay available right now on the market, and actually the most cost-efficient if you're going to use it. So, it's designed, it's allowed to amplify only CDR3 region or full-length region of all 7 chains in one reaction for all T cell receptor or B cell receptors. It also can be, you can start from RNA, from DNA, or even from cell lysate if you like, or single cell. It's really, it's work in any application. It's also, it can work with the cell-free DNA and extracellular exosomal RNA, if you're interested to really to study all these rare molecules which are derived from damaged tissue or from cancer or from tumors. Quantitative analysis you will facilitate it using so-called validator barcodes. It's concept similar to unique molecular identifier, UMIs, and it's very important when you analyze so complex population because you need to do error corrections and normalize all your number of the reads what you generate in next generation sequencing to number of molecules in your biological sample. Data analysis done by MIXR or platform, newest version. It is one of the most common publicly used, free actually for academic researchers. It's, if you look at number of publications, it's top software tool which you can use for immune repertoire profiling. It's not actually multiplex PCR, this assay. This assay based on the use of the mixture of different gene-specific primers, reverse primers and forward primers, and you're only doing 2 extension steps. First extension from reverse primer, follow extension from forward primer pool, and after that amplification using universal anchor primer. This one gives you uniform amplification of all CDR3 sequences or full-length region from RNA or DNA. Okay, so single-cell analysis, it's just briefly, it's about the same, but logic here following. When you do single-cell analysis, definitely a lot of people use 10x genomics or PARS or Rhapsody, for example, from Becton and Dickinson. But if your population of the cells is very rare, for example, you stimulate your T cells and label the cells with CD133 or any peptide tetramer or any other way. Population usually of these cells, it's very small. It's something like a couple hundred cells. Why do you need to run 10x genomics? You just sort the cells in 96-well plate, which already primers already for RSA already deposited here in each well with well-specific barcode. Put this plate in at -80 refrigerator, and when you need it, you can combine plates or analyze them at any convenient time for you. And it's allow you to generate exactly the same information. It's give you chain pairing information, full-length chain pairing information, and expression of 30 marker genes. Let's see how data looks like and how to analyze them. And they will give you only brief, just flavor about it, not very deep introduction. Okay, if you try to run this assay for 7 chains in, for example, PBMC, 50 nanogram of RNA, it will approximately amplify 100,000 clonotypes for TCR and 100,000 clonotypes for immunoglobulins. You can see it's rather balanced, so it doesn't hurt really to amplify both of them together and to get more comprehensive information. But the question is, are these data reproducible or not? For example, if you take another 50 nanogram of RNA and repeat this experiment, can you see the same clonotypes? The simple answer is no. And what is the problem? Problem here is like, here's experiment, the same experiment done in triplicates. And you can see here, all these bars, it's a variation in numbers what you detected in triplicates. And why it's happened, for example, here it's 100,000 clonotypes. From 100,000 clonotypes, 99,000 clonotypes approximately amplified only from one molecule. Definitely one molecule is not statistically significant. It's yes, no, yeah? It's like in single-cell analysis. You can see signal, no signal. And if you take another sample, it will be the same. It's really very random. Only statistically significant data start if you have at least in your population of RNA at least 5-10 molecules for each clonotype. In this case, the data you can see start to be reproducible and can be quantitative. Right now, okay, we can have idea how we are going to do quantitative analysis, how to identify activated clonotypes. I'll just give you an example, and this one's typical example. We collected multiple samples before vaccination and after vaccination using 2 types of vaccines. First of them, it's triple vaccine, tetanus, diphtheria, and pneumonia, and another is Shingrix against shingle infection. And we collected, you see, multiple time points and did expression profiling and TCR-BCR assay and single-cell assay. If you just run expression profiling, and this one is one of the products of our company, it's genome-wide expression profiling, 19,000 protein coding genes. It's also used the same logic, multiplex primers, 19,000 protein set of 19,000 gene-specific primers mixed together as a pool. And you can amplify it. You can see after 2 days after vaccination, you can see activation of innate immunity pathways. At approximately 1 week, you have strong activation of adaptive immunity pathways. If you look at the immune repertoire profiling, I show here in order not to really to make it very crowded, only from RNA and only 2 heavy chain for T cell receptor and B cell receptor. You see, it's very clear. Dominant response, you can see it's immune receptors, B cell receptors, antibodies. For both vaccination at the same time, approximately 1 week after vaccination, you can see strong activation, multiple, maybe 50, 100 of different clonotypes. And it's combination of expansion of these B cells and at the same time transcriptional activation, because majority of these clonotypes are probably plasma cells which start to produce immunoglobulin M. It's very quickly after 1 week of vaccination. You can see also activation of the T cell receptor about at the same time, 1 week after vaccination, which showed a clear indication that cooperative interaction between T cells and B cells in order to induce immune responses. The simple question will be, can we predict, based on the sequences what we identify here, if what antigen they recognize? And it's not so easy, because we collaborate with the company ImmuneWatch, it's a Danish company, and these guys are one of the best in prediction of the interaction between T cell receptors and peptides. And we ran first triple vaccine, we didn't find any match to the existing data sets. But for Chenchik vaccine, when we run this program, we see it's a machine learning tool, we show that it's majority of a lot of clone types what we identify correspond glycoproteinin from zoster virus, which is an integral part of this vaccine. So we show that it doesn't hurt really not to try to apply some bioinformatic tools and not to predict unknown epitope what recognized by any T cell receptor. If you just try to expand this type of the application for the typical examples what the people are doing when they try to discover T cell receptor which recognize some peptides which associated, for example, with the cancer, like illustrated here. And here's the typical examples what people are doing. It's a combination of experimental and bioinformatic strategies where the people try to associate it to identify cancer mutated proteins or overexpressed in the cancer proteins, or predict, for example, identify peptides using mass spec for eluted from the cancer cells, or even so-called black matter antigens which are derived from non-coding RNA, alternative splicing, line, or any repetitive elements. As a result, they generate a list of something like between 100 to 1,000 candidates. And right now the question is, which one of them has immunological properties? Which one of them can be used not to develop cancer-specific vaccines or develop, for example, cell-based therapies for treatment of the cancer? And the traditional way is just to take PBMC. It can be from clinical sample or it can be normal donor, synthesize a lot of bunch of peptides and do this type of the screen, which shown here on the next slide. And you can see here we try to optimize protocols in order to see how this technology works, what we have in our hands in this type of the protocol. What we done with design classical example cancer-associated antigens MART1, and we stimulate healthy PBMC samples with this MART1, grow cells for 2 weeks. At this time, what we take RNA from the beginning and DNA, and after 2 weeks, and just compare these two populations. So we can see here on the bottom left slide, you can rather easily identify, you can do quantitative analysis and can easily identify set of the clone types which are specifically, it looks like specifically expanded by MART1 peptide. After 2 weeks, usually what is standard protocol, it's you need to validate this data. In this case, you sort the cells based on the HLA tetramer staining, peptide tetramer staining, or expression of activation marker CD137, and doing single cell analysis in sorted cells in 96-well plate. And here's the results show how it looks the data in some sort of the heat map. And first 3 columns you can see hits we sorted the clonotypes based on enrichment after 2 weeks of stimulation with the peptides. And you can see here first column it's original population, second column it's DMSO control, it's non-specific stimulation with the same clonotypes. Here's the enriched clonotypes. I would like to put you notice that here's a level of enrichment here for the top clonotypes, it's several thousand-fold. So cells which are stimulated expanded in 2 weeks several thousand-fold. Can we just validate this data? And here's next 3 columns, it's validation data using CD137 marker, MART1 tetramer, and interferon gamma. And you can see here we have rather good correlation between this data, in spite that all these 3 technologies give you some sort of sparse data. It's not as robust as when you just do quantitative analysis of clonotypes by RSA. Actually, this one, last column, is interesting. It's prediction using the same algorithm what I described, detect, which shows also it's rather good. It's working rather well for MART1. And the main reason MART1 is extremely well characterized antigen. So that's why it's really some data present in the public domain databases. It's not always applicable for novel antigens, but for already known antigens described, at least you can find some match between data what we generated. So we selected some antigens and did some validation studies. Also about, in general, this MART1, it's easy target, but if you try to do some novel peptide, it's completely unpredictable, and it's like some sort of expedition where you don't know what will be result. It can be no at all any response. And this one is more complicated target. It's NY-ESO1. This one is also cancer-associated peptides, but not so abundant in PBMC as MART1. And you can see here we've done this experiment in triplicates, control triplicates, and stimulation with peptides. As you can see here, background here, non-specific expansion is significantly higher than in case of the MART1. So it's not so strong stimulator as MART1 peptide. And what is important, if you look at this reproducibility in triplicates, majority of the hits for control are overlapping in triplicates. But if you look at specific hits, for example, for NY-ESO1, you can see that they're unique for each triplicate. This one is rather easy to explain because number of the cells, which are naive cells, where these T-cell clones derived from, really extremely low. And if you take 1 million or 2 million PBMC cells, only maybe 1 or 2 cells has actually a T-cell receptor which can recognize this peptide. So definitely if you take another 2 million cells, it will be another T-cell receptor. So that's why you don't have any reproducibility, which sounds rather logical, and it's additional way to select really, to separate non-specifically amplified clones from peptide-specific TCR clones. Can we validate these results? And definitely, as I described, it's a classical artificial system, even introduced Rosenberg already probably more than 20 years ago, which based on the Jurkat reporter cell line, where you can clone any T-cell receptor or library of T-cell receptors, and K562 cells where you can clone HLA or complex HLA with the beta-2 microglobulin and peptide or spiked peptide. When you co-culture them together, if they interact specifically, GFP-induced, you can measure easily this regulation by FACS. And this one shows result of validation studies, and from 12 clone types what we selected from slides what I show you, 10 of them show very strong upregulation of GFP when in this co-culture experiment. So what we discussed? We discussed combination of this technology, bulk analysis, single cell analysis, and sorted cells in the 96-cell plate with downstream validation allow you to narrow down list of the candidates and to have confidence that it's correct really data. But can we scale up this? For example, as I mentioned in the beginning, usually typical experiment, you have 100 or maybe 500 candidate peptides. Can you do it for 100 to 500? Definitely this protocol probably it's not really the best strategy. If you have 10 postdocs, probably you can do it for 500 peptides and maybe rather good budget. Can we do it more scale up? And idea here following. We just test several technologies. There is rather hot area. It's a lot of publications about it. People describe many different strategies how to screen so-called one library against another library of peptide library against a library of T-cell receptors. And what we find is it's one of the most simple strategy, and it's really sounds rather logical, just to do this assay in microwells. And this one is technology developed by CellDome. And core of this technology, it's microwell. It looks similar, the same as 96-well plate, but in each well, you have 3,000 microwells. And each microwell, it's coated with antibodies, which can recognize, for example, interferon gamma or any other secreted protein. In this case, you load Jurkat reporter cells or CD8 cells or antigen-presenting cells, for example, K562. In each of them, you can clone, for example, in Jurkat cells, you can clone synthetic TCR library, or in K562, you can clone any synthetic peptide library of any complexity. And if you want any of these libraries, please contact us. We are a company which specialized in construction of any genetic library of any complexity, up to 200,000. So you can order any library you want of all of these molecules. You co-culture these two types of the cells in microarrays and do an assay. And so Seldom company is actually developed specific a special instrument to do it. And our next presenter, Guido, will tell you more about this instrument and how you can use it for this type of assays.